Welcome back, biology. And today uh, we're going to be talking about aerobic respiration. So we're going to take that pyruvic acid from yesterday and from glycolysis, and we're going to get all of the energy out of it. And we're going to use this thing called aerobic respiration. And this only happens when we have oxygen available. But aerobic respiration, which is a little bit different than cellular respiration is the whole process. Aerobic respiration is just this process with oxygen. Is going to take is going to do two parts. The Krebs cycle, which we call the citric acid cycle, because it involves the molecule citric acid, and then the electron transport chain. And both of these parts are important to get all of that energy out of glucose to get all that ATP. Right now, we're only at 2 out of the 38 ATP that we need to produce. So we need to produce 36 ATP through this process. Before we jump into it, we need to talk about NADH. It is important because it is used in the electron transport chain to make ATP. So whenever you see us making an NADH or an FADH2, these molecules are high energy carriers. They're going to take these electrons to the electron transport chain. We're going to use them to make ATP. The Krebs cycle, the electron transport chain, and chemiosmosis can only occur if oxygen is present. We kind of mentioned this before, but oxygen is really, really important to the process. There are some slight differences between prokaryotes and eukaryotes. In prokaryotes, the reactions of the Krebs cycle and the electron transport chain take place in the cytosol and this fluid portion of the cell. This is because they don't have a mitochondria. But in eukaryotes, we do these kind of reactions inside our mitochondria. If you remember, these mitochondria were actually probably early prokaryotes that were eaten and not absorbed because they had these kind of properties to produce ATP. All right, so now we're taking this pyruvic acid. It diffuses across the mitochondrial membrane into this matrix, into this yellow on here, fluid-filled space inside of the mitochondria. So anytime you see matrix, think space. The mitochondrial matrix is just the space on the inside of the mitochondria. Another important molecule that happens is this pyruvic acid goes in here, it reacts with coenzyme A, which is an enzyme here, to create acetyl-CoA. And acetyl-CoA is important. It is it is what is needed to start the process of the Krebs cycle, the citric acid cycle. So we lose one carbon, we turn into this 2-carbon molecule. So we lose a CO2, but we gain an NADH that is going to be used in that electron transport chain later. All right, you should have a copy of this, the Krebs cycle walkthrough. I'm going to go through step by step the process of the Krebs cycle, and then as I go through it, just fill out your walkthrough, and then you'll have kind of a better graphic organizer than if you were trying to write it down in your notes yourself. All right, step 1, we take this 2-carbon molecule of acetyl-CoA, and we combine it with a 4-carbon molecule called oxaloacetic acid. Here. This produces a 6-carbon compound called citric acid. The amount of carbons are kind of below these molecules. There's only a couple, oxaloacetic acid and citric acid, that I need you to kind of think about and know. The rest of them we're just going to talk about the number of carbons they have and what's produced in each step. So first step, we've made citric acid. This is why it's called the citric acid cycle. It's because this is a cyclic event where citric acid is kind of a very key player. All right, number 2, the citric acid releases a carbon dioxide molecule and a hydrogen ion atom. This creates a 5-carbon molecule here, 1, 2, 3, 4, 5. The lost hydrogen combines with the electron to convert NAD+ to that NADH and a free hydrogen ion here. This is going to go play in the electron transport chain and give us lots of energy there. So this is kind of like an investment. This is going to turn into really good energy. All right, in step 3, we're going to take this 5-carbon, we're going to release another CO2 molecule and a hydrogen to form for the first time, a 4-carbon molecule down here. We're also taking another NAD plus and turning it into NADH, and we're taking an ADP plus a phosphate and turning it into ATP. And remember that ATP is energy. So we're creating more energy each time we go around the citric acid cycle here. For step 4, we're taking that 4-carbon molecule and we're releasing another hydrogen to form a different 4-carbon molecule. This also takes another energy carrier, FAD, and turns it into FADH2. And FADH2 is also going to go to the electron transport chain and be converted into energy. And then step 5, we're going to take that 4-carbon molecule, we're going to release another hydrogen atom to make oxaloacetic acid. This is going to take this NAD plus and make another NADH. Now it's important to note that each time we go through this, we're breaking down half of a molecule of glucose. So if we go around twice, we'll break around a whole molecule of glucose. So the efficiency of the Krebs cycle, like we said, 1 glucose gets broken down every 2 cycles. So that means when we do 2 cycles of the Krebs cycle, we get 2 ATP produced per glucose molecule. And the question is that more effective? And the answer, of course, is not yet, because we got 2 from glycolysis and we got 2 from the Krebs cycle. We're still missing 34 ATP somewhere. To get these ATP back, we have the electron transport chain. So far, we've made 8 molecules of NADH, 2 molecules of FADH2, and we need to get the energy back from those. And we need to reconvert those back to NAD plus and FAD so that they can be used again in the Krebs cycle in the future. They're enzymes, they don't get used up, but they need to be recycled. The electron transport chain is going to take these and convert them into ATP. They're going to make ATP out of them. So the electron transport chain is a series of proteins, of molecules, that transfer electrons from a higher state to a lower state. And in eukaryotes, this is embedded in the mitochondrial matrix, in those folds called cristae. We have lots of folds of the cristae so that we can have lots of room for the electron transport chain, so we can make a lot of energy. If you're a prokaryote, if you're a very simple bacteria or archaean, you are going to do this just in your cytosol, and you'll have these proteins just in your cytosol. So, how do we make ATP? When we have NADH and FADH2, and they release their hydrogen atoms, they reconvert back to those NAD pluses and FADs. When these hydrogens are removed, their electrons are in a high energy state, and they need somewhere to go. And we're going to harness that using the electron transport chain. I have a second walkthrough for the electron transport chain for you guys. It works exactly the same as the second one, but instead of them appearing, they kind of peel off. It was a little easier to give you guys space this way. So in step 1 here, we're going to take NADH and FADH2, and it's going to give up their electrons to the transport chain. NADH gives up its electrons at the beginning, converts back to NAD+, and FADH2 donates further down the chain and becomes FAD again. These molecules also give up their hydrogens. And it's important you see these hydrogens going through here. Those hydrogens are going to play an important role at the end of the electron transport chain. All right, step 2. We're going to take those electrons, we're going to pass them down the chain, both here and here. Each time they move from one molecule to another, they lose energy. Now that energy is going to be used for something. So in step 3, we're going to take that energy and we're going to pump protons from the matrix, from the inside here, to the outside. So each time an electron kind of moves through this chain, we pump one here, we pump one here, and we pump one at the end here. So 3 different areas we're pumping these hydrogen ions out. What this does is creates a concentration gradient and an electrical gradient. Now we have way more H pluses out here and way more E minuses in here. In step 4, this concentration is going to run the ATP synthase down here through that process of chemiosmosis. It's kind of the same process we see in photosynthesis. Protons are going to move from their high concentration out here down through ATP synthase, and that energy of electrical and concentration gradient is going to turn ADP into an ATP plus phosphate into ATP. So now we've created energy. Finally, oxygen down here is the final electron acceptor. It is going to accept the electrons, it'll accept these extra protons coming in, and when they all combine, they will form 2 molecules of H2O here. So oxygen is really important, final electron acceptor. Without it, our protons and our electrons inside the cell would get too high. They wouldn't be able to continue to work the electron transport chain. It would shut down, and you wouldn't be able to make any more energy. So without it, the electron transport chain stops working. And in your muscle cells, if you're working really, really hard and you've used all the oxygen, it stops working in muscle cells. And then you only have to use glycolysis, but then you have to use that lactic acid fermentation, and that causes muscle cramps and fatigue. All right, a real quick summary of cellular respiration. The first step is that glycolysis. Glycolysis is all about taking glucose and turning it into pyruvic acid. We use 2 ATP to do it, but we make 4 ATP. So altogether, we net a positive 2 ATP from glycolysis. And then in aerobic respiration, we're making 2 molecules of ATP from the Krebs cycle, and we're taking pyruvic acid and converting it into molecules of carbon dioxide and water. And then after the ATP, kind of the electron transport chain, we make 34 more from those NADHs and FADH2s for a total of 38 ATP. So for each molecule of glucose, we put in 2 ATP and we get 38 back if we use the full cellular respiration. That's a pretty good return on investment, and that's quite literally the energy that runs everything that you do in R. So this is the conclusion of Chapter 7. I hope the walkthroughs helped. I'll try to figure out how to link those in the description so that if you're watching this and don't have them, you can get them. But if you have any questions, let me know. Have an excellent rest of your day, and I'll see you in the next video.